Hello, this is Dr. Didar Alam, Assistant Professor of Biochemistry and Biotechnology Department, North South University. Today we will study Lecture 6, the remaining part of the Lecture 5. We were studying enzymes. So today we will study what are the factors influence or affect the enzymatic activity. So today we will start from enzyme concentration. So rate of the reaction directly proportional to the concentration of the enzyme when pH, temperature, substrate concentration are constant. So if we plot rate of the reaction against enzyme concentration, so the reaction rate will be directly proportional to the concentration of the enzyme, but the condition is we need to keep pH, temperature, and substrate concentrations are stable. So look at here in this plot. So the rate of the reactions or the product concentration is low because of the absence of enzyme. If you increase the concentration of the enzyme, the product concentration will be increased, and if you increase the concentration of the enzyme more, the product concentration will be increased. If you continuously increase the concentration of the enzyme, and if you keep constant supply of the uh, concentration of the substrate, then the rate of the reaction will be increased. So there is a chance uh, if you uh, stop the continuous supply of the substrate, so the all the enzymes will be occupied, all the enzymes will be occupied with the substrate, and substrate will be converted to product. And there, if there is no substrate, if you increase the concentration of the enzyme, the rate of the reaction will not be increased with the increase of the concentration of the enzyme. So to get the rate of the reaction higher, or to get the rate of the reaction increased with the increased concentration of the enzyme, you need to continuously supply the substrate. So what are the inhibitors? There are some inhibitors that are called enzyme inhibitors. These are the molecules that interact with the enzyme. This interaction might be temporary or permanent, and this reduce the rate of the enzyme catalytic reaction or prevent enzyme to work in a normal manner. So they are usually specific, and they work at low concentration, and they block the enzymes, but they do not usually destroy it. And many drugs and poisons are inhibitors of enzyme in the nervous system. So what are the types of inhibitors? Competitive inhibitor, there are three types: competitive inhibitors, non-competitive inhibitors, uncompetitive inhibitors. So competitive inhibitor, this compete with the substrate molecules for binding with the active site. So competitive inhibitors compete with the substrate molecules to bind with the active site, so that these are called competitive inhibitor. And this inhibitor's actions is proportional to its concentration. So they have close similar structure with the structure of the substrate. So what about the non-competitive? It inhibits the binding. It inhibits by binding irreversibly to enzymes, but not at the active site. So they binds the allosteric site, the other side of the enzyme, rather than the active site, and these are not influenced by the concentration of the substrate. So this competitive inhibitor can be overcome with the increased concentration of the substrate, but this will not, because it not binds with the active site. This binds with the allosteric site. So uncompetitive inhibitor, this binds to the enzyme substrate complex to stop the enzyme from reacting with the substrate to form product. As such, it works well at higher substrate and higher enzyme concentrations. That substrates are bonded to the enzyme. So uncompetitive inhibitors works by binding with enzyme substrate complex. So if you increase the concentration of the enzyme and concentration of the substrate, so that enzyme substrate concentration will be higher, and then this uncompetitive inhibitor will bind with the enzyme substrate complex, so inhibition will be higher. So here is the example. So this is the active site of the enzyme, and substrate bind here, and this is the enzyme substrate complex, and this is the product. So competitive inhibitor, they have similar structure with the substrate, and they bind with the active site of the enzyme and compete with the substrate. So this keeps substrate not bounded with the enzyme. And a non-competitive inhibitor, they don't bind with the active site, they bind with the another site, allosteric site. So this is the table that summarizes all the types of uh, inhibitions. So it's very important to remember this table. So look at here, the substrate bind with the active site. This is the enzyme. An inhibitor compete to bind with the active site. So there, inhibitor bind with the enzyme and it forms enzyme inhibitor complex, and inhibitor bind to free enzyme only and competes with substrate. So increasing substrate can overcome this inhibition by inhibitor. So not non-competitive inhibitors, there is a different site in the enzyme, this is called allosteric site. So inhibitors bind with the free enzyme, or look at here, inhibitor can bind with the free enzyme making enzyme inhibitor complex, and then substrate can bind to produce enzyme inhibitor substrate complex. So the reaction rate will be decreased or stopped. So here inhibitors can bind free enzymes or enzyme substrate complex. So increasing substrate cannot overcome the inhibition because here inhibitor bind with the allosteric site. So uncompetitive inhibitor, here inhibitors can only bind enzyme substrate complex. So increasing substrate favors the inhibition by inhibitors, because if you increase the substrate concentration, it will form enzyme substrate complex and inhibitor bind with the enzyme substrate complex. Okay. So another type of inhibition is feedback inhibition. Feedback inhibition regulates the activity of the most of the enzyme, where the pathway can regulate by feedback inhibition. So the end product of the pathway binds at the allosteric site of the first enzyme, so altering its structure of the pathway and shutting it down the pathway. So this is the formation of thyroid hormone from first to last, and this is the hypothalamus, and increased level of thyroid hormone signal hypothalamus to stop synthesis of thyroid stimulating hormone. So when product is abundant, active site are full and enzyme activity drops. When product is used up, inhibition is reduced and more product is produced. So this is another example of feedback inhibition, where look at here, this is the step 1, step 2, step 3. So this is the enzyme substrate complex and intermediate substrate. This is the product for this reaction but substrate for this reaction. Product for this reaction, substrate for this reaction, and this is the end product. So this end product will bind the first enzyme at the allosteric site and will inhibit the binding of substrate to the enzyme. So look at here, the major digestive enzyme. These are carbohydrate digestion, the enzymes for protein digestion, enzyme for nucleic acid digestion, enzyme for fat digestion. So salivary amylase, pancreatic amylase, and maltase. This the salivary amylase produced in salivary gland, work in mouth, and work in neutral pH. Pancreatic amylase produced in pancreas, work in small intestine, and work in basal pH. Maltase produced in small intestine, released in small intestine, and work in basic pH. And pepsin, this is a protein digesting enzyme. This produced in gastric gland and work in stomach, so in acidic pH. Trypsin produced in pancreas and work in small intestine in basic pH. Peptidase produced in small intestine, work in small intestine in basic pH. Nucleic acid digestion, nuclease works produced in pancreas, and both nuclease and nucleoside is produced in pancreas and released in small intestine, work in basic pH. And fat lipase produced in pancreas and released in small intestine, work in basic pH. So you can see only pepsin that work in stomach is in acidic pH. So because stomach has acidic pH and small intestine has basic pH, so the enzyme that work in stomach will work in acidic pH, the enzyme that works in small intestine work in basic pH. So examples of cofactor, this is the enzyme and active site, and here is the prosthetic group. This is one type of cofactor tightly bounded with the enzyme, and coenzyme, these are loosely bounded with the enzyme, and these are also cofactor. So we studied organic compound and inorganic compound. That means cofactors are two types, organic and inorganic. If it's organic, it's called coenzymes or prosthetic group. So If if the organic compound bound enzyme with the enzyme tightly is called prosthetic group, or loosely it's called coenzyme. So, catalyst of biological system. Enzymes are a highly effective catalyst, and they are highly effective, commonly enhancing the rate of the reaction by 10 to the power 5 to 10 to the power 17 fold. And enzyme catalyzed reactions are characterized by the formation of enzyme substrate complex, and enzyme substrate binding occurs in a product on the enzyme called active site. So, enzyme substrate reactions are characterized by the formation of complex between enzyme and substrate. This is called enzyme substrate complex, and substrate bind in a orchid on the enzyme is called active site. The function of an enzyme and other catalysts is to lower the activation energy delta G for a reaction and thereby enhance the rate of the reaction, and the equilibrium of a reaction is unaffected by enzyme. So how catalysts affect the reaction rate? So look at here. So enzyme affect reaction rate, not the equilibrium. So a simple enzymatic reaction might be written like enzyme substrate complex, enzyme product, enzyme product. So to understand the catalysis, we must first appreciate the important distinction between reaction equilibrium and reaction rate. So the function of a catalyst is to increase the rate of the reaction, and catalysts do not affect the reaction equilibria. Any reaction such as substrate to product can be described by a reaction coordinate diagram, a picture of the energy changes during the reaction. So this is the reaction coordinate diagram where free energy has been plotted against the reaction coordinated. So this is the reaction coordinate, this is the free energy, and this is the substrate. The substrate in the beginning of the reaction is called in the ground state, and this is the transition state, that is the activation energy, and after the formation of the product, this is the energy state of the product. This is called ground state. So this is the energy change of substrate to product, and this is the energy change of product to substrate, and this is the net free energy change which can be achieved from the difference between this one to this one. So biochemical standard free energy changes. To describe a free energy change of a reaction, chemists defined a standard set of the condition, like temperature should be 298 degree and 98 Kelvin or 25 degree centigrade temperature. Partial pressure of each gas will be 1 atmosphere or 101.3 kilopascal, or concentration of the each molecule or each solute should be 1 molecule, and express the free energy change for this reaction system as delta G, the standard free energy change. So the starting point of either the forward or reverse reaction is called the ground state. The contribution to the free energy of the system by an average molecule, substrate or product under a given set of the conditions. And this is the transition state at the top of the energy hill. There is a point at which decay to the substrate or product state is equally probable, and it is downhill either way, and this is called transition state. So the equilibrium between S and P, substrate and product, reflects the differences in the free energies of their ground state. In the example shown in figure here, the free energy of the ground state of the product is lower than that of the substrate. So delta G for the reaction is negative. The equilibrium favors for substrate to product. So I repeat, the free energy change from transition state to ground state for product is lower than the free energy change from transition state to product is transition state to substrate is higher. So here is the substrate, here is the product. So this is the free energy change will be negative, and then the reaction will favor from substrate to product. So the transition state is simply the fleeting molecular moment in which events such as bond breakage, bond formation and cleavage and charge development have proceeded to the precise point at which decay to either substrate to or product is equally likely. So this is the transition state. And activation stage, the delta G, the differences between the energy level of the ground state and the transition state is the activation energy. The rate of a reaction reflects this activation energy. A higher activation energy corresponds to a slower reaction rate. So reaction rate can be increased by raising the temperature, thereby increasing the number of molecules with sufficient energy to overcome the energy barrier. Alternatively, the activation energy can be lowered by adding catalyst. Catalysts enhance the reaction rate by lowering the activation energies. So enzymes are not exception to the rule that catalysts do not affect the reaction equilibria. The bidirectional arrows in equation 6 here makes the point any enzyme that catalyzes the reaction substrate to product also catalyzes the reaction product to substrate. The role of the enzyme is to accelerate the interconversion of substrate and product. The enzyme is not used up in the process and the equilibrium is unaffected. So what are the reaction intermediate? Any reaction may have several steps involving the formation and decay of the transient chemical species called reaction intermediates. A reaction intermediate is any species on the reaction pathway that has a finite chemical lifetime longer than a molecular vibration is equal more or less is equal to 10 to the power minus 13 seconds. So when a substrate to product reaction is catalyzed by an enzyme, the enzyme substrate and enzyme product complex can be considered as a intermediates. Even though substrate and products are stable chemical species, the enzyme substrate and enzyme product complex occupy values in the reaction coordinate diagram. Additionally, less stable chemicals intermediates often exist in the course of an enzyme catalytic reaction. So the interconversion of two sequential reaction intermediate thus constitute a reaction step. When several step occurs in a reaction, the overall rate is determined by the step with the highest activation energy, and this is called the rate limiting step. The rate limiting step is the highest energy point in the diagram for interconversion of substrate to product. So, what is the biological significance of activation energy? Activation energies are barrier to chemical reactions, and without such activation energy barrier, complex molecules would revert spontaneously to much simpler molecule forms, and the complex and highly ordered structures and metabolic processes of cells do not exist. And over the course of the evolution, enzymes have developed lower activation energy selectively for reaction that are needed for cell survival. So that's all from the lecture 6. See you in the lecture 7.